Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing you another really fascinating guest today who is helping to create a better tomorrow on many unique fronts. Today, we have the honor of being joined by Dr. Stephen Moran, who is Global Program Head of Neuroendocrine Tumors and Other Radiosensitive Cancers for Advanced Accelerator Applications, which is a Novartis company. Stephen is also a member of the Oncology Development Unit leadership team at Novartis. Prior to joining Advanced Accelerator Applications, Dr. Moran was global head of Novartis strategy, and there he played key roles in defining the company's strategy, helping to prioritize critical actions needed to deliver on various parts of the mission to help discover new ways to extend and improve people's lives. He has led numerous strategic initiatives across the various units of Novartis, including in gene therapy, in RNA therapeutics, precision medicine, and digital strategy. Prior to Novartis, Dr. Moran served strategic assistant to the CEO, a position that he held for 2 years. And prior to that, he was associate principal at McKinsey Company, serving as a leader in their healthcare practice. There he focused on health system sustainability, research and development strategy, and economic analysis of clinical interventions across various disease pathways. Dr. Moran holds a Bachelor of Arts and a Master of Science in Biochemistry from University of Cambridge in the UK. He also did undergraduate work at MIT, and then also received his doctorate from Oxford in biophysics, where he lectured on various topics, including thermodynamics, quantum mechanics, electromagnetism, all applied to biology. Dr. Moran has also been a guest lecturer in strategy at University of St. Gallen, International Executives MBA, master's and bachelor's courses, and received the Young Investigator of the Year award in 2006 from Biochemical Journal. A lot of really interesting themes to get into today. We're honored to have him. Dr. Stephen Moran, thank you so much for taking time to come on the show today.
Thanks for having me, Ira. Appreciate it.
Yeah, it's a really fascinating set of topics we're going to be getting into today. I took a little time in preparing for this show, looking at some of your early work when you were doing your PhD, and reading. Glancing through papers like mechanical explanation of RNA pseudoknot function and program ribosomal frame shifting, mechanisms of translocation, spring and ratchet systems. Biophysics is a completely fascinating domain to me, applied to both genes in human biology, but also a lot of your focus was on viral pathology. Take us for a few minutes through a little bit of the early days, if you would, how you got interested, not just in the world of biology, but biophysics in general. I think that'd be an interesting way to start things off here. What would we
Wow. Well, first of all, thank you, Ira. I don't think anyone's been ever so prepared for an interview that I've spoken to, so appreciate you digging into it. No, it's a long time ago now, but yes, biophysics was a great love of mine, still is. The physicality of it, that, life is a physical thing, where you can look at it, and you can see it, and you can understand its dynamics and the forces and the movements, and push and pull of the cell and the energy of it. It's just absolutely fascinating insight into what makes life tick, the physics, the physical principles, the biophysics of things. And so yes, my research started on viral replication mechanisms, things like SARS virus at the time, which is related to to sort of coronavirus things and fundamental replication mechanisms, and how the RNA of viruses can hijack the cellular machinery of the cell and reprogram it, and taking the electron microscope and taking pictures of the thing and looking to see what it does. And so that was really fascinating work, and understanding these molecular machines has been a theme for me. Yeah.
Excellent, excellent, and obviously fits perfectly into sort of everything you're doing at Advanced Accelerator applications. But one other place before we get to AAA, before we go there, I want to stop at one other place, and of course, a little bit about, as I mentioned in the bio, you've had this important strategy role over the last several years, working not just for one, but two CEOs of the Novartis organization, looking at strategy across really all domains of the company, everything from the generics business to gene therapy to RNA. You've been involved in digital health, in the China strategy, precision medicine, and so forth. I mean, I could go on for a while here, but two really interesting things sort of pop out here. Number one, of course, Novartis as a company, it's moved well beyond sort of how we've thought about traditional big pharma in the past, of just sort of discovering, developing, registering, and commercializing sort of small new chemical entities. But also, you have served as this important change agent, let's say, in big pharma, which is always very difficult to change. We spent 100 years or so working on small molecules, and you're doing all sorts of really non-small molecule cool stuff that is really, obviously everyone agrees this is going to be the future. Talk a little bit about your role, because I know you've been quoted in a lot of papers in terms of pharmaceutical strategy, scripts and in vivo and so forth. Went through some of those articles. Talk about a little bit of that role as well, if you would, and how it's sort of you've been able to move and help move Novartis into some of these unique sort of cutting edge areas of biopharma.
Yeah, thanks. So I spent 4 years as the head of strategy for Novartis, sat on the executive committee, and reported to two CEOs. And when I joined Novartis, it was right in the middle of our portfolio swap with GlaxoSmithKline and others. And so the great sweep of my time in that position with the company has been the move of Novartis as a conglomerate structure, as a holding company with multiple business unit independent P&Ls, from animal health and vaccines and generics and eye care and innovative medicines and oncology, to a focused medicines company. So we span out or swap those businesses, and then with that capital, then we made the decision to reallocate the capital into high-end, cutting-edge technology platforms. And the thesis there really was, partly Novartis was criticized for two things. One, its scale is too big, and it's too broad. It covers too many disease areas, and that's a legacy, thing. And the question was, could we turn those two weaknesses into a strength? And the idea was, if we could get cutting-edge platforms that require high capital intensity over a long period of time, that would play to our scale. It would be, why be a big pharma company? What's the point, right? And the number one strategic principle really is synergy effects. You have to have synergy effects. Otherwise, you may as well take each business unit, the cardiovascular franchise, or whatever, and spin it out as little biotechs, right? The overhead costs off. So you're looking for synergy effects. So can we take our scale and invest it to build these technology platforms that could then simultaneously cut across multiple disease areas? So you have that synergy effect, and then you also have the scale capital effects that essentially builds a moat around those technologies that makes it very difficult for others to penetrate. And so this would be our definition of a platform technology, something that can cut across disease areas. You can build a moat around it with big long-term capital investments. And it has reproducible innovation. The concept can be applied multiple times in different ways. And so we started that with the cell therapy investments. And so the CAR T technologies, we were the first company to bring the CAR T Kymriah to market in the US. We pioneered that. Curative type technology has never been seen before. This kind of big ticket price, one-off payments for curative type things is a new thing, right? But massive manufacturing challenges and scale and infrastructure associated with that. And then we did a similar thing with gene therapy. So we bought really the world's first big gene therapy product, Zolgensma, the world's most expensive drug, to market, again, curative type sort of effect in spinal muscular atrophy. We've seen that before. And then we had a third technology platform, which was radioligand therapy, which is what I'm working on now. And we also invested again into RNA technologies for public health, like vaccine-like kind of use in cardiovascular disease, which we think is the forward looking. If you think about where health systems are going in terms of prevention over the long term, we made a big bet, $10 billion bet by the medicines company to go in that direction, which was much criticized at the time. We'll talk about that. But of those four platforms, the radioligand therapy has always been my favorite platform. It best meets the criteria for what a platform is, that I described in terms of reproducible innovation and capital investment. The supply chain of these radioisotope atoms is part of the product. So you have to deliver the product within a certain time window where the half-life decay of the isotope hasn't gone too far and the energy is too low. It's not too high. You have to hit that window, and you have to take the order and then produce the thing with the isotope and be able to ship it logistically. That's tough, right? So, and then the concept is relatively simple. It's like an antibody drug conjugate, but it's like a radio drug conjugate. You can have different vectors, and you just put a single isotope atom on. So it's reproducible. Right, and so that was always my favorite one, and so then at some point I decided to put my money where my mouth is and try and step in and bring this forwards, partly because it married to my biophysics background, and so that was a unique opportunity for me to take that on.
Absolutely. And so, so enter advanced accelerator applications, and you have this sort of broad mission, reimagining nuclear medicine and cancer care, and going through some of the materials. You talk about that this is not your grandparent, well, not your, I tell you, your great grandparents' nuclear medicine anymore. If we go back to the days of Röntgen and Curie and so forth, but nonetheless, we've been using radiotherapy for over a century now. But this is much different. You are, as you were mentioning, you're developing these targeted radioligand therapies, specifically oncology. Talk about, if you would, and we'll get into some more of some of the targets and the manufacturing themes as well. But talk a little bit about this this topic of theranostics, basically where you're marrying this very unique form of nuclear precision medicine, which we don't really think about nuclear diagnostics, but it's a diagnostic and a therapeutic together. Talk a little bit about what that means.
Yeah, no, it's very cool. It's at the basic level, it's you take a tumor targeting molecule, the ligand. It could be a peptide, it can be small molecule, it can be a small antibody fragment, whatever you want. You attach a radioactive atom to it, and then it binds to the tumor cell like a targeted therapy, and then the radiation damages the DNA of the cell, causes DNA breaks, which either slow replication or induce apoptosis. And that's it. And in some ways, the concept is doing what small molecule targeted therapies did for chemotherapy back in the day, for what this would be doing for beam radiation, and going from something that is sort of approximate to sort of an organ or a tissue level, and taking it to a very precise molecular cellular basis.
And you are, you have responsibility for our neuroendocrine tumors. The company is also focused on the prostate. Can you talk a little bit about why these novel moieties are useful in these solid tumors? I know that's one of the things, even with the antibodies and the CAR T and so forth, done outstanding in sort of the bloodborne cancers, but been kind of tough with the solid tumors. Talk a little bit why sort of this technology maybe opens the door a little more for some of these smart tools to work on the solid tumors that you're working on.
Yeah, well, I think it's got a lot of promise now. We started with the first radioligand therapy, which was Lutathera, and that targeted GEP-NET, neuroendocrine tumors, relatively rare disease, growing. And we brought that asset to market. It's doing very well. And then we thought, well, we'll have more of that. And so we brought forward the PSMA targeting agent. Lutathera was the SSTR receptor targeting agent. Now we brought the PSMA targeting agent. But the mechanism is very similar. In both cases, we use the same radioactive atom, a single atom of lutetium-177, it's a beta-emitting radiation isotope. So it's the same atom on both. It's just this time, the PSMA is targeting into prostate cancer, into receptors on the prostate cell. And then that worked as well. So we had an improvement of median overall survival, read out positive last year, and that asset, Pluvicto, is now launched in the US. And what's quite, again, is that reproducibility of the innovation is what we've been looking for, right? And so the exciting thing here is that it works, seems to work. The technology works in two totally different cancers, which raises the thesis, could this technology be a sort of universal mechanism of action? Is it that, the simple physics of radiation through space makes the problem simpler? So now maybe the specific internal cell biology of one tumor type to the next doesn't matter. As long as we can get the atom to the cell surface for long enough, maybe it works, right? And so that's what's sort of unique about it. It's so simple, and now we're talking about physics. You can't really argue with it. We're not talking about the inhibition of an individual signaling cascade and the adaptations of the cell therein. Now we're just talking about radiation through space to the DNA damage, and you can't argue with physics. And now this becomes a physics problem, not a biochemistry problem. And so then what we're dealing with then is a whole new mechanism of action that we haven't seen before in oncology.
Yeah, no, that's extremely exciting. I mean, when you think about that dimension of it, and some of, once again, using an older tool, revisiting it, reimagining it, as sort of the mission statement says, and now in 2022 now, taking advantage of it. So that's really exciting. Stephen, as you were mentioning, earlier on, obviously, aside from creating that unique moiety or combination moiety that you have to take into account, obviously, we don't want too much radiation, we don't want too little, and we have to make sure that we can get to that patient in a certain amount of time. We can't come across the country, but a certain distance. I noticed recently you created a new production facility specifically, I think it was in Indianapolis, for these novel radioligand therapies. Could you talk just a little bit about, we hear obviously, what we've heard a lot in the recent decade about, obviously, the antibodies coming online and that we don't have enough capacity and we need to build more and so forth. What are some of the other issues that come into play with regard to the scale up, the ability to produce enough of these novel products in the future? And at the same time, we get into the smart theme a lot on the show in terms of, the smart home and the smart health and what have you. Any interesting smart manufacturing related tools that are interesting that help you specifically when it comes to these novel radioligand therapies?
It's a big question. As I said, the manufacturing process itself is uniquely part of the product, physics of the product. And so, manufacturing radioligands involves the production of pharmaceutical grade radioisotopes, as well as the chemical synthesis to bind these to the targeted moieties so they're ready to use, ready to administer patient dose. And so each dose is produced for an individual patient. So this just-in-time kind of methodology done at a molecular level. And that timing is crucial because you've got very limited shelf lives, few days. And so one of the main challenges is coordination of that radioisotope supply. These come from rare earth elements using nuclear reactors. And so I never thought Novartis would own nuclear reactors, but we do now as part of our supply chain. So that adds more to our risk management process, give our risk management colleagues a headache. But they need to be sufficient to supply the precursor element as well as access to the reactors. It's not that common. And so we've been trying to approach these challenges, including sort of M&A to augment our in-house capabilities and long-term agreements with suppliers for both the precursor and the production. And then we're constantly evaluating new production methods for those isotopes, for different treatment settings, and thinking about as well, do we need to put RLT manufacturing sites in proximity to treatment centers for the delivery of the final product? As well, of course, then robust automated logistical planning processes. And that would all have to be adapted potentially by the type of isotope we're using as well. So this is a whole infrastructure build. And again, on the theme of platforms, I like that complexity, even though it can give us headaches, because every headache we get and we overcome is a learning in our bag.
Oh, yeah, absolutely. Yeah, it also brings to mind, one of the topics we've gotten into on previous shows is this principle of digital twin, and the ability to, once again, really in the virtual world, figure out how this is all going to happen before, as you were just saying, hey, deliver me the thorium or whatever it may be, and I got practice with the live material. So yeah, I think there's a lot of really interesting things happening behind the scenes there that are probably pretty exciting for a pharma company. So and someone like you that has this background. So that's really exciting. It's interesting, as you were talking about nuclear plants and sort of the supply chain here. A couple of months ago, I had Dr. Najat Makhtar, who is the Director General and the head of nuclear science at the IAEA on the show. And she's a biologist as well. And, she didn't want to talk about nuclear power or anything like that. She wanted to talk about biology. And one of the things she was really excited about was something called Zodiac, which stands for their Zoonotic Disease Integrated Action Initiative. And as you were pointing out, these isotopes are really good at killing cancer cells, novel mechanisms, also really good at killing all sorts of other nasty bugs. With your background, obviously, in sort of the viral pathology area, what excites you beyond oncology? Obviously, you have a lot to work on with oncology, but as you sit there with these unique tools, what else gets you excited in terms of human health and potentially infectious diseases?
Well, for me, maybe it's a little boring. Don't know if it's boring or not, but for me, it's prevention.
Okay.
And the science of prevention and population health. And so, for oncology, the big win is obviously the earlier you can catch the cancer, the easier it is to treat. And I think so. There's so much more we could do in terms of the science of screening populations, whether it's detection of circulating tumor DNA in the blood, or other detection techniques and genetic profiling, and take those technologies coupled with information technology and AI and do large scale population screening. And I think the benefits one can get from that are huge, because there is the risk that we try to tackle cancer once it gets too late, that it's like a runaway train.
Yeah.
You're just fighting to stand still. But if you can do the prevention, it's a huge difference. And that's why, I was a big advocate as part of our strategy at Novartis to move after this company called the Medicines Company, which is an RNA-based technology that can be used in a preventative way, where instead of taking statins every day, which is a very low compliance in the population at large, you can inject this RNA therapy, once every 6 months when you go for your regular 6-month checkup, and it dramatically reduces your cholesterol. and so there's no compliance issue, super safe. And so you can apply that over massive populations, and the benefit to the health system, economically and in terms of patient lives, is just enormous. It swamps, everything else you could do probably in biomedicine. So that's what I'd love to see us getting into: early prevention, and then coupled with treatment methodology like radioligand therapy to intervene early.
Yeah, no, that'd be huge from the prevention perspective. Yeah, clearly, it's exciting to see that you're already thinking ahead in terms of not only have a novel technology here, but that's really an untapped space. So it's exciting to see that potentially Novartis getting into that as well. Stephen, are you learning any interesting things while we're thinking of still on radiation? Anything about sort of the reverse, as you have to handle, or your manufacturing has to handle these unique moieties that are not normal? Anything on sort of the radiation resistant side of things? We've talked with folks from NASA on previous episodes that obviously they're very concerned with radiation, and they study extremophiles and tardigrades and things like Deinococcus that are resistant to all sorts of radiation. Any interesting things you're learning along the way regarding to the radiation resistant side of things?
It's an area of investigation. This technology is very young, and so we don't fully understand how resistant or not cells are getting. And one of the interesting things about radioligand therapy, a bit like our cell therapy or our gene therapy technologies, at the moment they're a little bit of a one and done type treatment. Not quite one. And usually we do maybe 4 cycles over a period of 6 months or so, but then we stop. So we don't keep on treating chronically like you would do with other cancer medicines or other medicines. It's sort of a blast, and then we stop. And that's partly, because we've just had general concerns about how much radiation is too much radiation to expose, what are the long-term effects, and then because of a young technology, you need to follow it over time. But we have had 5 years safety data now for our first radioligand therapy. So even though it has these connotations of radiation, because it's done in such low doses, this cellular level doses, it's order of magnitudes below what beam radiation is. And so what we haven't explored properly is, well, what if we retreat? What if we keep on treating? Are patients becoming resistant? Are they becoming overexposed? Or if the receptor is still there, can we keep pulsing over time and keep the cancer in check? That's a great unanswered question for us.
It's going to be an interesting set of coming years as you explore these novel solutions. Really very interesting stuff. Stephen, I wanted to just now ask you a little bit. Obviously, when I read your bio at the beginning, you got your doctorate in biophysics, but you lectured on various other themes in terms of thermodynamics, quantum mechanics, electromagnetism. Once again, all applied to biology. And as here's a perfect example that you've been talking to us about these novel approaches using radiation and some of the novel therapeutic moieties. But I'd love to get your perspective on where else, looking out, and maybe Novartis might not have investments in these areas yet, but things that once again get you excited. There are other, I won't say any bad names, but there's other pharma companies that have put some of their bets in so-called electroceuticals. And once again, coming back to the zap, maybe not radiation there, but zapping with electricity. Quantum biology has become a hot topic for us in very early days. And then of course, thermodynamics, heating tumors to a point of apoptosis and so forth. With sort of your broader biophysics hat on, take us a little bit to the future. What else do you see transpiring maybe over the next 5, 10, 15 years that can move us for more of these biophysical approaches to human health?
Oh my gosh, I don't know how broad I can go. I think
take your time.
I'm a little focused on the radioligand therapy at the moment, to be honest. This sort of absorbs out of mind, and maybe let me start there. Maybe about where I think that could go next.
Okay, sure.
Because it is very exciting. I think, as I was saying, the proof of concept that the technology can work in two totally different cancers raises the premise, well, could it be a universal mechanism for multiple cancers? And so strategically, where do you go next? And our philosophy has been, well, if you're privileged enough to have in your hands a totally new mechanism of action that hasn't been used in biology before, and that's very rare that happens, why don't we try and take it into some of those cancers that have proven intractable to the other historical mechanisms of action and see if it works there? So that's our posture, and then we kind of look to say, well, these are the cancers that have been the most difficult to tackle to date. Which of those are radiosensitive? And so then you get into things like glioblastoma, so brain tumors. It's been so intractable. Small cell lung cancer, very super aggressive lung cancer. And so, that's where we're trying to take the technology next, into these big, high unmet need cancers. And I think beyond that kind, our technology, well, first of all, there's the possibility to go into cancers as well, into more broad-based disease with the therapy, and we've talked about like inflammatory diseases. We attack the white blood cells with the radiation, and I think beyond that, I think the nanotechnologies are very exciting. And can we develop nanotechnologies that go into the brain that can be combined with magnetic fields, etc., and pulses to trigger explosions in specific, a bit like the radioligand therapy or your ADCs, but maybe using nanotechnologies coupled with precise magnetic fields, etc., to carve out tumors or other diseases. DNA scaffolding and the RNA technologies, building off, and I think the combinations as well. So, the cell therapies that we talked about, with the gene therapies, with things like the targeting radiotherapies I've talked about, and the RNA technology. Part of our thesis for the long term was, if we can build up expertise in these difficult technology platforms, we can also then start to combine them. And I'm looking for synergy effects, right, as the great strategic principle of what's the point of a big pharma company. If we have these things in-house and we keep our ways of working good, can we start to synergize and use gene therapies to express receptors in a cell that then can bind the best of our radioligand therapies to target in, combine, modify the cells, the CAR T technologies in different ways with the gene therapies, maybe activated with magnetic fields or radiation pulses and things like this. So yes, bringing the physics and the biophysics together with the biology, and hopefully at some point it starts to all merge, and we have such a command over these technologies that you can essentially reprogram disease almost at will. That's where they would go eventually.
Really exciting. I love the combination concepts, and then as you're just mentioning with the sort of the non-cancer, the autoimmune diseases, and we've touched a lot on sort of the rare diseases, whether it's rare autoimmune conditions that they repurpose cancer drugs for. It clearly, it all fits into to your basket. It's going to be a very promising future there for you in the coming years, and you have not just a great portfolio, but a lot more that you're going to be building. So they're very exciting times.
The strategy is to bring it from both the two perspectives that I described. So the one is to bring these biophysical methods, these gene therapies, these combinations that we could have in-house at Novartis to tackle, the most aggressive diseases as they've progressed, and some proportion will always need to be treated at some point, with a scientific and high-end technology approach to the science of prevention, and you attack disease from both ends, at scale. And it's a company like a big pharma like Novartis has the scale to be able to take on population health challenges, and then the scale to combine technologies at the end of the disease life cycle and try and get it from both ends.
Absolutely. Stephen, what, anything else hot for 2022, conferences you're going to be presenting at, places where we can hear you, meet you, talking about advanced accelerated applications, anything else that I might have missed, that please take the floor on that one.
Yeah, we'll be at the major conferences, later this year, ESMO, etc. So yeah, we'll have a high presence at all the major conferences, really. So look forward to seeing you there. And then, the trends that we'll be talking about and trying to explore is, from a radiopharmaceutical point of view, so as I said, the move into new indications. Then we'll be talking about the move into different types of isotopes that we can put in there. There's a lot of talk about alpha-based therapies like actinium-225. So at the moment, most of the technology is based on beta emissions.
Okay.
So electron kind of based emissions. Alpha-based emissions are larger alpha particles,
yep,
are more ionizing. So, we can combine these things in different ways. And then combination therapies. So combinations, again, of RLTs with immunotherapies, so PARP inhibitors and other bispecific antibodies or binding domains such as the DARPins. Investigation of diagnostic isotopes like copper-64 is a big theme. And there's lots of debate about the dosimetry. I mean, one thing we haven't talked about is there's a very unique property of these radioligand elements where you can image the molecule in the individual patient as well as use it to treat. So not only can you diagnose the extent of the cancer and where it is, i.e. you treat what you see in this person, you can also do what's called dosimetry measurements. So in the PET scanner, you can actually measure the intensity of the binding in the image. So you can measure for the individual patient how much of the molecule is going to the healthy tissues, how much is going to the tumor, and then what is the intensity of the radiation dose, equivalent of what would be a dose in a chemical term, into the tumor. You can measure that for an individual patient. And you basically get a perfect pharmacokinetic, pharmacodynamic profile in the image with a time course, so you can see it coursing through in very rich levels of detail that you would never get with a normal molecule.
Yeah.
You can. It's a very powerful technique for drug development, and how you maximize that component of the technology and use it, that's a big theme as well. So all of these themes you see us discussing at the conferences, etc., over the coming year. Yeah.
Excellent. Very exciting times. I'm rooting you on. I love biophysics. I love the combinatorial theme. I really think you're in such a sweet spot for leveraging the investments that you've made early on and seeing the potential of some of these novel tools. So it's really exciting. For everybody that is going to be listening to this particular episode of the show across the various podcast networks, or who'll be watching it on our YouTube channel. Again, you've been listening to Dr. Stephen Moran, Global Program Head, Neuroendocrine Tumors and Other Radiosensitive Cancers for Advanced Accelerator Applications, part of the Novartis organization. He's also a member of the Oncology Development Unit Leadership Team at Novartis. Stephen, I want to thank you so much for taking the time to walk us through these very complex themes. I obviously thank you for everything you're doing for human health and the unmet medical needs. And as we like to say on our show, thanks for helping to create a better tomorrow through what you're doing. It's a very exciting platform. Really wishing you the best with all of it.
Hey, thanks a lot. I appreciate it. Thanks for the interest and the time today. It's very exciting to talk about it with you.
Good seeing you. Be well.